Tobias Buchborn is my name. I'm currently with the Institute for Psychopharmacology at the ZT in Mannheim, Germany. And in our institute, we do research on the clinical applications of psychedelics, but we are also as much interested in the mechanism of action. So how do they do what they do? And we believe if we truly want to understand the therapeutic implications of psychedelics, it's important that we also know what it is they actually do and what is their essence. And this is what we're going to talk about today, not so much about therapy, but rather what is it they're doing on the neuronal level. When we very simplistically ask, what is it they are doing? What is it we are knowing so far? What is most robust? We know that psychedelics activate the serotonin 2A receptor, and this is then the protein entrance door, basically, for the psychedelic experience. And we know it because when you pharmacologically block this receptor, then they wouldn't be able to do their trick anymore. You would then ask, we just heard it in the last lecture as well, where do these receptors expressed? And the answer is actually they are expressed all over the body. Serotonin 2A receptors can be found all over the body, but the densest expression, do you see the laser pointer, is actually in the visual cortex, so in this area, and the other largest expression is in the frontal cortex, which is the highest region of the cortex, the high associative cortex. And here the prelimbic cortex is particularly important, and we will focus today a lot on this prelimbic cortex. So if we then move on to EEG and MEG, particularly EEG is the method that is being used the most in human research, particularly if you add in the research from the 50s, then what is most established is that wherever you're looking, if you look in the frontal cortex, in the parietal cortex, wherever you're looking, locally you would find a desynchronization in the EEG. And this has been shown for psilocybin, LSD, mescaline, EEG, so this is really something that is highly replicated. And desynchronization on a local level actually means that you have a pool of neurons which normally would fire together. They would release the glutamate, for example, at the same time, and then you would find very strong amplitudes. But if they do not fire together anymore, you lose amplitude and you lose entrainment of the neurons. They all do whatever they want to do. So this is what desynchronization means. You're aware of the fact that there's all kinds of network analyses, default mode network analyses, but we will not talk about this today. We want to go beyond the cortex, and also we want to go beyond what EEG can measure. If we want to dig a little bit deeper, we actually have to move away from animal research, from human research, and enter animal research, and not even in living animals, but rather taking out the brain, making slices, and doing in vitro electrophysiology. If you apply psychedelics into this prelimbic region, so if you remove the brain from the animal and then put psychedelics into the prelimbic regions, we find that particularly pyramidal cells of the layer 5 of the cortex respond to psychedelics. They would become more easily excitable, and if you repeatedly stimulate them, they are less refractory, so they do not habituate. They would fire again and again, although the stimulus is not new anymore. And there's quite very nice and extensive research on how this happens in this electrophysiological model. And the idea is here that there is just too much glutamate. It's basically a rain of glutamate. So the current story, the current narrative is that the psychedelic experience is a hyperglutamatergic state of the cortex induced by serotonin 2A receptors. The problem is this cannot be validated in humans because there's no method for it. We also cannot do it, but there are very recently methods in animals which allow us to do a bridge from this EEG to this model of hyperglutamatergic state. And this bridge we want to cross today together. And the bridge we are using is called in vivo fibre photometry. In vivo fibre photometry can be imagined like an EEG. But unlike EEG, you can measure in a sleeping animal, in a head-fixed animal, but also in an animal that is walking around. Unlike EEG, you cannot only measure the cortex, but you can also go beyond the cortex. In fact, you can go in whatever area you're interested in. So even if you do the right genetic targeting, you can actually even pick the neurons you're interested in. And what I find most sexy about this method is actually EEG can only measure voltage. This method can measure everything, at least in some future. You can measure voltage, calcium transients, glutamate release, serotonin 2A receptor activation, second messenger production, metabolic changes, and hemodynamics. And the list of things you can do with this method is ever growing. So instead of just measuring voltage, you can actually do all kinds of other recordings while the animal is asleep, while the animal is walking around, while the animal is under psychedelics. And this allows us to bridge the EEG findings with those findings we have in the in vitro model. Just a short note on how this works. This is actually an optogenetic technique, and it works via genetic engineering. We use non-hazardous viruses. Viruses are like vehicles. We can insert a gene into it, and then we can insert the virus into those cells we are interested in. Then they start, a gene is basically a cooking book. So then the cells which are infected with the virus start to produce a new protein. This protein has the same function as endogenous proteins, but every time it fulfills the function, it would emit a light. And then we can basically measure from the outside with the fiber whenever glutamate is binding, for example, or whenever serotonin is released, and so forth. This is roughly the technique. And we record from two sites, what we're going to talk about today. Number one, we record from the prelimbic cortex. We talked about it because this is where a lot of serotonin 2A receptors are. And here we insert a glutamate reporter. It's called GluSNF. So whenever glutamate is raining, the reporter would tell us. In addition, we also record from the thalamus, a certain thalamus which is part of the midline thalamus, and the midline thalamus is very important for filtering. It's in a very important interface between the subcortical world and the cortical world. It's like an entrance door. So the thalamus, as you know, is a gatekeeper to consciousness. That's how it's called. And here we would express GCAMP. GCAMP is a measure of how excited the thalamus region is. And in order to target the right cells, we actually did antibody staining. So we would basically have a look into these regions, and we don't want to measure the prelimbic cortex as a whole, but we only want to measure the layer 5 pyramidal cells. This is where the dense labeling is here. So we use antibody labeling, and then we can just measure where is the peak of the serotonin 2A receptor expression and how much is the distance from the midline. And then we can basically, when we do our surgery, just move from the midline to the part where the layer 5 pyramidal cells are, and then we are quite sure that we measure from the right cells, or at least from the right layer. Okay, this is an example of a representative recording. This is actually a recording from one animal over 3 hours across the different states of waking. This is very deep anesthesia. If the anesthesia is very deep, the cortex by times is totally active, and you see here basically glutamate release. This is what you see. This is medium anesthesia, low anesthesia. Here the animal wakes up, and here you see the animal freely walking around. And sometimes we apply saline in between, and sometimes we apply a psychedelic. And what is quite interesting, already tried just from a basic neuroscientific perspective, is that during anesthesia, there's a lot of glutamate, and all cells fire together. That's why you have these high amplitudes. The more the animal is awake, the more the synchronicity is getting lost. So as you can see here, this is freely walking. Neurons are more diverse in their patterns, and they would not fire together as much as they do in anesthesia. In our research, we work with 3 psychedelics. We work with LSD, psilocybin, and quipazine. Today, we will focus on quipazine. The reason you probably have not heard of quipazine, it's used a lot in animal research because it's behaviorally quite potent, and it's more easy to tickle out relevant stuff if you use quipazine. But we'll also speak a little bit about LSD, but most of what I showed you is based on quipazine. It's a serotonergic psychedelic, a serotonin 2A agonist, as much as psilocin and LSD are. Which results? This is glutamate release within the prelimbic cortex. This is a baseline before we inject our psychedelic drug. Then the red vertical line is where we inject quipazine, and you can see with the naked eye that something is going on here. It takes only a few minutes, actually, before the amplitudes are drastically decreased by the drug, and this tells us that there's indeed a desynchronization of the glutamate release in the prelimbic cortex. And this is something we know from EEG, but only in terms of voltage, not in terms of transmitter release, actually. And these colors are very beautiful, but it's not about the beauty here so much. It's rather each of the different colors represents one window, and I would do a sliding window analysis, and of all windows, I would do a Fourier transformation. Basically, the Fourier transformation tells us about the spectral architecture of the signal. So is there actually oscillatory signal in there? This is what it tells us. And you can see when we just focus on the post-drug, at the beginning, there are high amplitudes, and if we look at the yellow Fourier transformations, there's actually quite some delta activity going on, but the longer the drug effect moves on, so the more we approach red, the more the architecture is lost. As you can see, all of this power is lost here, and this tells us that there is a desynchronization on a spectral level, and it affects the deep, slow oscillations. This is basically a way of quantifying, so we would determine the area under the curves for all of these Fourier transformations. This is just meant to do the statistics, and as you can see here, the amplitudes are significantly reduced after the application of the psychedelic, and this tells us there's this desynchronization. We also measured the peak frequency in this low frequency area, and there's actually an acceleration going on after the psychedelic drug, and both actually has also been shown in vitro in this in vitro model of Marek and colleagues. So this is actually very nice that we can one-to-one find what they showed in vitro now in vivo. This is a spectrogram. I also like this picture a lot because it sums up things. It's basically what we've seen before. Here you see the frequencies. Here you see the time as the signal goes on. The red vertical line is where the drug was injected, and as you can see, here's a lot of redness, and this redness tells us there's a lot of low frequency power here, but the longer you move to the right, the more the redness vanishes, telling us that there is a desynchronization. There's actually, probably you have noticed it, there's also another signal up here, and this is actually, can anyone guess what this is? It's actually arterial pulsation, because we measure light, and the light trespasses arteries, and these arteries constrict and dilate, and this is how we can actually measure the dilation of the arteries within the prelimbic cortex. And what we can see here, and we will have another look on the next slide, that the drug indeed increases the arterial pulsation of the arteries within the prelimbic cortex, something that hasn't also not been shown before in this way, at least. This is actually a quantification of it, so we would use mathematical modeling to extract the signal from the background signal. This is what you see here, and then you can do basically your statistics, and then you see that there is an increase in frequency in the dilation of the arteries within the prelimbic cortex. This is just a side finding, but why not capitalizing on it? It tells us something about the physiology of psychedelics. This is the next picture, and it looks almost, if I switch back and forth, like it was the same, but now we are in the thalamus. So as you can see in the thalamus, there's of course also arteries, and these arteries in the thalamus react in the same way as the arteries within the prelimbic cortex. This tells us, you also see this increase in frequency here, and this tells us that the effect the psychedelic has here has something to do with something that is beyond the arteries, so most likely they increase the pulsation of the heart. This is what we're seeing here. Okay, this is the neuronal signal in the thalamus, and I would like to show you as a comparison how the prelimbic cortex looks like. Because in direct comparison, you see that compared to what we see with the glutamate release in the prelimbic cortex, the effect of the psychedelic on the thalamus is way more severe. We will later have a look at the spectrogram. You will see that the signal and the synchronicity of the signal is almost completely gone. And we would quantify this in the very same way. We would do our Fourier transformation. And if we now have a look at these yellow bits, so after drug injection, you see there is still a very nice oscillatory architecture. But the more we move to the red part, the more there's just noise. So the quibosine makes the thalamus stop being synchronous, up to a level of complete noise, actually. And this is, again, a quantification. As you can see, I forgot the stars here, but this is highly significant. And also there's an acceleration here. But as the signal-to-noise ratio in the thalamus massively drops, we need to be careful with the interpretation of this acceleration. But the desynchronization is obvious. This is actually my favorite picture of everything I'm showing to you. Here in animal research, we also use placebos, which is a saline injection, so it's an isotonic salt solution. So we, of course, compare whether the injection per se has an effect. And as you can see here, if you inject your saline, there's nothing going on with the thalamic activity, but with quipazine, there's complete asynchrony. I need to tell you that not all animals show such a severe effect, but it's really something, an effect that stood out so massively, so it was popping out. So I've been doing research for quite a bit already, but I've never seen something so obvious. So it was really a beautiful finding. And here are statistics from the saline versus quipazine. And as you can see, both in the glutamate, with regard to the glutamate release in the prelimbic cortex, but also with the thalamic activity, there is a desynchronization. Yes, it's Eastern, but this is not the reason why I'm showing you this slide. Actually, I added this slide because I wanted to have this sink in a little bit, because as you know, all of the story, all of the narrative is about the cortex. So this is partially biased by the fact that EEG is used a lot, and EEG can only tell something about the cortex, but these findings raise the question of chicken and egg. What is first? And this, of course, is the holy grail of science, and I will not be able to answer it today, but we can ask the question, what is first? Is the thalamus breaking first, or is it the cortex that is breaking first? And in order to answer this, we will not answer it, we can approach it. We can actually look into the interaction of both. So we can measure both things in the same animal at the same time in a highly synchronized manner, and then see whether the communication of both areas is intact or whether it is affected. And the easiest thing you can do is actually a cross-correlation. Here you see that before you apply the drug, the cross-correlation between both signals, the glutamate release in the prelimbic cortex and the thalamic activity, is near 1. So they are perfectly correlated in terms of their linearity. But after the drug application, this sinks to half of it. And this can also be analyzed again with the sliding window analysis. And you here see that there's actually a drastic drop, whereas we are at around 1 at the beginning, we almost end up with 0 in the end. So this means the glutamate release we still see in the frontal cortex is completely disconnected from what the thalamus is doing. Okay, this is another measure. It's sort of similar like a correlation, but it's called coherence. And then the coherence is not so much about the linearity, but the question is rather, does the cortex and the thalamus, do they share the same oscillatory architecture? So is the way of oscillating, is it the same? And you can look at this sliding window analysis of the coherence, and as you can see, in baseline, there is a very nice coherence, particularly in the low frequencies. You again see the heart up here, and after quipazine, especially in the later phases, there's actually no coherence, again showing that they just stop communicating, these two areas. Yeah, again, our sliding window analysis, and the coherence here drops to near 0, actually. This is the last parameter I would like to show you. It's called mutual information. Let's have a look at the joint distribution of the amplitudes before. So before we give the psychedelics, this is here the interaction between the prelimbic cortex and the thalamus. So before we give the drug, they share amplitudes across a whole variety of values. So you see there's a very steep joint distribution. But after the drug, everything both have in common is around 0. So after the drug application, the only thing in both areas share is actually noise. And this is actually a way of depicting the parameter I want to show you now, which is mutual information. Mutual information is the question, if I know the glutamate signal in the cortex, can I predict the thalamic activity, or the other way around? And as you can see, before the drug injection, this is possible to a certain degree, so you can predict the one parameter based upon the other, but afterwards this prediction is not possible anymore. Okay, there are all kinds of these parameters, so I want to spare you any more of them, but I would like to end the results part with this chicken and egg question by doing such an analysis here. So the question is, which of these areas breaks first? So quipazine is being injected here at the vertical black line, and then we need to find a way to quantify which one is first to break. And it's actually not so trivial, so we use multiple approaches and then make an average across these different approaches. I would like to introduce you to one. We would basically fit a linear regression to the baseline phase and would then, so the pre-drug phase, and would then elongate it across the drug phase. And then we determine basically when is the first time that the peak in the signal lies under this linear curve. And this is then the breaking point. And when we do this analysis, and we have now found it in all of the animals we have done so far, the answer is the thalamus breaks first. And this is interesting, again, because it moves away from the narrative that the cortex is the initiator and that the cortex is the most relevant thing. So we find it's not only that the cortex is more severely affected by the psychedelic, we also find it's affected before the cortex. Okay, we are at the end already, so I wrap up what we have talked about today. What do we know? We know from human EEG that no matter in which area of the cortex you look into, we have a local desynchronization. We know from rat research in vitro that psychedelics increase the excitability of pyramidal cells of the layer 5, particularly in the prelimbic cortex. But there has never been a bridge. There was no bridge to this. And we tried to bridge this in our research. And we have found that in the prelimbic cortex, there is indeed a local desynchronization and acceleration of glutamate release onto deep layer pyramidal cells, which is very much in line with the idea that glutamate is a key driver here in psychedelic drug action. Also, we find in the midline thalamus, so we didn't look at the thalamus as a whole, we looked into the midline thalamus. We also found a local desynchronization and the desynchronization that occurs in this thalamic nucleus, the midline thalamus, paraventricular nucleus actually, that the desynchronization is way more severe than the desynchronization we have in the cortex. And it is also that thalamus breaks before the cortex. Some limitations here. Number one, we use fibre photometry. So the recording is based upon a fibre which emanates from the brain. And the diameter of this fibre is 400 micrometer. Of course, the regions we are recording from, the thalamus and the cortex, are bigger than 400 micrometers. So it's possible that if we had recorded from a neighboring subpopulations, that the timing would have been different. So it's not that we can say for sure that the thalamus is always the one to break, but just based upon what we have so far. But we would have to take multiple recordings from the same site to really nail this down. So there's no causal implication here. Also, we need to have in mind most of the research I've shown you, or all of the research I've shown you today, is with quipazine, and quipazine is not selective like all psychedelics are. It has a particularly high affinity for serotonin 3 receptors as well. Other psychedelics do not share this property, so it's possible that these serotonin 3 receptors also play a role. This is something we will have to figure out in the future. Some discussion of our findings with regard to what other people have done. In this in vitro electrophysiological model, which I referred to in the beginning, it has actually been shown that if you lesion the midline thalamus, then the psychedelics are less active in the prelimbic cortex. So it seems that psychedelics indeed need the midline thalamus, which is in line with what we show here as well. Another thing is that our data also fits the thalamocortical filter model of psychedelic drug action, which has been introduced to the field by Vollenweider, as you probably know from schizophrenia research. He adopted it into psychedelics, and our results are pretty much in line with the lack of filtering, because the impact the thalamus usually has on the cortex is not there anymore because they stop communicating. This means the cortex becomes more independent of this filter. and this makes it prone, theoretically at least, to other information crashing in from the limbic system or from primary sensory areas. This is just an outlook. First results with LSD, prelimbic cortex, glutamate release, and just to show you that LSD does the same trick. We still need to go into the thalamus, but with regard to the glutamate release, we have replicated this now for this drug as well. Thanks to those people who are involved, particularly Markus Meinhardt, the head of our working group, Rainer Spannagel, Wolfgang, thanks for their support. Particularly I want to thank as well to Sung-Kyu Lim and Violetta Bartos, who are my students and whom without I wouldn't have been able to collect all these doing a lot of surgery, doing a lot of recordings, really a lot of work. So thanks to them, and thanks for you for your attention.